Have you ever really stopped to wonder what's actually happening inside your head? I mean, right now, as you're listening to me, billions of tiny events are unfolding at lightning speed just to make it possible. So today, we're going to dive into one of the most fundamental processes in the entire universe, the spark of thought itself. I mean, think about it. Your every thought, every feeling, every single movement, what are they actually made of? At the most basic physical level. It's a huge question, and it really gets right to the heart of who we are. And the answer? Well, it's both super simple and absolutely mind-blowing. They're all made of electricity. Yep, tiny, ridiculously fast electrical sparks. Your entire conscious experience is basically a symphony of these little electrical pulses, a biological light show happening inside your skull right now. And the conductor of this whole symphony, the star of our show, it's this incredible little cell right here, the neuron. So let's dive in and find out exactly how it creates that amazing spark of life. Okay, so before a neuron can actually fire off a signal, it has to be ready. It kind of just sits there in this state of quiet, poised potential, just waiting for the right cue. Think of it like its default setting. This is what we call its resting state. The scientific term for this is the resting membrane potential, and it sits at a very, very specific number, minus 70 millivolts. So what does that mean? It just means the inside of a resting neuron is electrically negative compared to the outside. A great way to think about it is like a tiny charged up battery, just holding its energy, waiting to be used. And this diagram really shows you why it's negative. See, on the outside, you've got this sea of positively charged sodium ions. But on the inside, sure, you have positive potassium ions, but they're mixed with these big, bulky, negatively charged proteins. That's the A- you see. And those proteins, they're too big to get out. They're stuck. So their negative charge is trapped inside, making the whole interior more negative than the outside. This imbalance, that's the key to everything. All right, so how does the neuron keep this delicate balance going? Well, it's an active process, and there are three main players. First up, you've got the sodium-potassium pump. This thing is constantly working, pushing more positive ions out than it lets in. Then you have these leaky potassium channels that let even more positive charge escape. And finally, a few leaky sodium channels let just a little bit of positivity trickle back in. It's this perfect coordinated dance between all three that holds the voltage right at that magic number, minus 70 millivolts. But of course, a neuron doesn't just sit around doing nothing forever. It's constantly being bombarded with messages from thousands of other neurons. You can think of these messages as little nudges. Some are pushing it towards firing, and some are holding it back. So let's see how those nudges actually add up. So these little nudges, these whispers before the shout, are called graded potentials. And they basically come in two flavors. You've got your go signals. Those are excitatory. They make the neuron a little more positive, nudging it closer to firing. Then you've got your stop signals. Those are inhibitory. They do the opposite, making the inside even more negative, holding it back. It's this constant tug of war between go and stop. So let's say just one or two other neurons send a little go signal. It's not nearly enough to cause a big event, right? But the voltage inside our neuron, it creeps up just a little bit. It's a small, temporary nudge towards action. Okay, but what if a bunch of go signals all arrive at the same time, or one after another really fast? Well, they start to add up. Their combined power creates a much, much stronger nudge, pushing that voltage even higher. We're getting closer now. And then it happens. The go signals finally overwhelm the stop signals. All those little nudges add up, and the internal voltage crosses this critical point of no return. That threshold is minus 55 millivolts. The second the neuron hits that exact number, everything changes. Because crossing that threshold triggers an unstoppable chain reaction, a massive electrical spike called an action potential. This isn't a nudge or a whisper anymore. Nope, this is the shout. This is the fundamental unit of all communication in your brain. This graph right here, this is the signature of a thought, and the whole thing happens in about 2 milliseconds. It's insane, and it's an all or nothing event. It's like a light switch. It's either all the way on or it's off. There's no in between. You can't have half an action potential. So let's walk through this step by step. Okay, step one. The second our neuron hits that magic threshold of minus 55 millivolts, thousands of tiny molecular gates just fly open. These are the voltage-gated sodium channels, and they're like these super sensitive floodgates that are just waiting for that exact voltage. So, the floodgates are open, what happens? Remember all that positive sodium that was packed on the outside? It comes rushing into the cell. I mean, it just floods in. This is called depolarization, and this rush of positive charge is so massive and so fast that the voltage inside just skyrockets from -55 all the way up to a positive 30 millivolts. Right at the very peak of that spike, two things happen at once. First, those sodium channels slam shut. Done. And at the exact same moment, a different set of channels, the potassium channels, they swing open. Now all the positive potassium that was inside the cell rushes out, taking its positive charge with it, and that causes the voltage to plummet right back down. We call this repolarization. Now those potassium channels, they're a little slow on the uptake, a little slow to close. So for a split second, a little too much positive charge escapes, and the voltage actually dips below its resting state. This is called the undershoot. And during this brief reset period, that hard-working sodium-potassium pump gets back to business, restoring the original balance and getting the neuron back to -70 millivolts, all reset and ready to fire again. Okay, so everything we just went through, that is the bedrock of neuroscience. It's what's been in textbooks for decades. But, science is always evolving, and a recent discovery has just thrown a completely unexpected plot twist into this classic story. See, for decades, the fundamental belief was that neurons just figure this out on their own, that the ability to fire an action potential is just an innate property of being a neuron. That they're basically born ready to go. But a team at Stanford, led by a researcher named Brad Zucchero, found something that totally flipped this idea on its head. And in his own words, the result was completely unexpected. What they did was they isolated developing neurons in a lab dish, totally on their own, away from all other cells. And what happened? The neurons never learned how to fire. They just stayed silent. It turns out neurons can't do it alone. They need a partner. And that partner is a type of cell called glia, which honestly were kind of dismissed for years as just being passive support cells. But get this. These researchers found that glial cells release an essential chemical signal. That signal is basically the instruction manual that tells the neuron how to build those critical sodium channels, the very gates that kick off the whole action potential. Without help from glia, a neuron never learns to speak. So why is this such a big deal? I mean, this discovery doesn't just change a diagram in a textbook. It opens up completely new ways of thinking about brain health, about development, about disease. It's this incredible reminder that even the most fundamental processes in our bodies are part of this bigger, interconnected community of cells. And this really matters. I mean, think about it. Problems with how neurons fire, this dysregulated excitability, are at the root of diseases like chronic pain and epilepsy. So understanding that these glial cells are actually key players, well, that could unlock totally new kinds of treatments. It just makes you wonder, right? What other fundamental secrets, what other secret conversations are happening in our brains right now that we just don't know about yet? The spark of life, it seems, is way more complex and a lot more collaborative than we ever imagined.
Welcome to the deep dive. Today we're tackling something truly mind-bending. The human brain. I mean, just think about it. Billions of neurons, all firing, communicating faster than you can blink. How does that even work? How do these tiny cells manage this incredibly complex dance? That's what we're digging into. We're going to try and decode the brain's hidden conversations. Our deep dive today, it starts small like with the electrical buzz inside one neuron. Then we'll see how they talk to each other across these tiny gaps and how the whole system changes over time. And finally, we'll get into some really cutting-edge stuff tools that let scientists actually control these signals. We've got some great sources lined up: Wikipedia on the Hodgkin-Huxley model, very well mined on action potentials, overviews of synapses, research on neuroplasticity and pain, stat pearls on resting potential, and even a paper on optogenetics. Quite a mix.
Yeah, it's a fascinating collection, and our goal here for you is to weave these threads together. We want to build a clear picture, starting from, the basic physics of a nerve impulse, all the way up to these sophisticated biological tools that are changing medicine and how we even think about intervening in the brain. It's really about seeing how that fundamental science, that deep understanding, leads to pretty revolutionary applications. You might be surprised where the connections lead.
Okay, let's dive in. Segment 1, that electrical spark. How does a neuron actually fire? The first thing to grasp is this all or none idea. It's like a light switch, right?
Exactly. It's either fully on, full blast, or it's off. No dimmer switch, no half firing.
Before it fires, it's just sitting there
Uh,
in a resting state.
But it's not passive. Think of it like a coiled spring, just full of potential energy, ready to go. Inside, the neuron is negatively charged compared to the outside, usually around, say, minus 70 millivolts. That specific voltage is key.
And that charge difference comes from ions, right? That's sodium, potassium.
Precisely. You got positive ions like sodium, Na+, potassium, K+, calcium, Ca2+, and negative ones like chloride, Cl. Plus, there are these bigger negatively charged proteins stuck inside the cell. The cell membrane is picky, though. It controls who gets in and out. At rest, it's pretty tough for sodium to get in, and potassium is mostly kept in, but chloride can move more freely. Those big negative proteins, they're trapped inside.
Okay, so it's negative inside, ready to spring.
Yeah.
But how does it maintain that state? How does it keep that tension?
Well, that's where the real work happens. There's this amazing molecular machine called the Na plus K plus ATPase pump. It's constantly working, using energy, to pump 3 sodium ions out for every 2 potassium ions it pumps in. It's actively building up these concentration differences, lots of sodium outside wanting in, lots of potassium inside wanting out.
So the pump sets the stage.
It sets the gradients, yeah. But the resting potential itself is mostly generated by potassium ions leaking out. See, the membrane has these special channels, K+ leak channels, that are open at rest. So potassium follows its concentration gradient and flows out, taking positive charge with it, making the inside more negative.
Okay.
If potassium was the only thing moving, the inside would get down to about minus 90 millivolts. That's its equilibrium potential, where the chemical push-out perfectly balances the electrical pull-in. But the membrane isn't perfectly impermeable to sodium. A tiny bit leaks in, making the inside slightly less negative. So it balances out around that minus 70 millivolt mark we mentioned. It's a dynamic balance.
Right, that coiled spring tension, so the neuron's primed. What pulls the trigger? What makes it actually fire that action potential?
Good question. It starts when some kind of stimulus arrives, maybe a signal from another neuron. This causes special sodium channels, voltage-gated ones, to open up. And because there's way more sodium outside and the inside is negative, sodium ions just flood into the cell, positively charged ions rushing in.
Making the inside positive now.
Exactly. That's depolarization. And here's the critical part. If enough sodium rushes in to push the voltage past a certain point, the threshold, then bang! The action potential fires completely, all or none. This ensures the signal zips down the axon, the neuron's long transmitting cable, without fading out, like dominoes falling consistently, all the way to the end.
No weak messages allowed.
Oop.
Makes sense for reliable communication.
Yeah.
So it fires. Then how does it reset itself so quickly?
Right, it needs to get ready for the next signal. 2 main things happen. First, repolarization. Those voltage-gated sodium channels snap shut, stopping the influx. At the same time, different voltage-gated potassium channels open wide. Now potassium rushes out, taking positive charge out, bringing the inside back down towards negative.
Okay, back to resting.
Almost. There's a very brief moment, the refractory period, maybe a millisecond or so, where the neuron just can't fire again no matter what. It's like a quick reset button. This ensures signals are kept separate and distinct. Keeps the timing right.
It's incredibly precise. And this whole mechanism, this ionic dance, this leads us to the Hodgkin-Huxley model, doesn't it?
Absolutely, a landmark achievement. What's amazing is they figured this out back in 1952 using the giant axon of a squid, believe it or not.
A squid.
Yeah. And they developed these mathematical equations, these nonlinear differential equations, that perfectly described how the changes in ion flow create the action potential. They treated bits of the membrane like electrical components, capacitors, resistors.
So they built a mathematical model of the scark.
Pretty much. And it was so accurate, so fundamental, it won them the Nobel Prize. It really laid the groundwork for understanding all nerve impulses, not just in squids. It's the bedrock we build on.
Incredible. Okay, so we've got the spark within the neuron. But one neuron firing alone doesn't do much. The brain's power comes from the connections, right?
Yeah.
The network. How do they pass the message along? How do they bridge that gap between cells? That's where synapses come in.
Exactly. Synapses are those specialized junctions where one neuron talks to the next. And there are basically 2 main flavors, electrical and chemical.
Let's start with electrical. You said they're like a direct wire.
Yeah, that's a good way to put it. They have these things called gap junction channels that physically connect the inside of the presynaptic cell, the sender, to the inside of the postsynaptic cell, the receiver. So the electrical current, the ions, can just flow directly from one cell to the next. It's super fast, virtually no delay.
Wow. Instantaneous. Where would you need that kind of speed?
Think about escape reflexes, like crayfish needing to flick its tail instantly to get away from danger, or a goldfish darting off. Those use electrical synapses. Also, coordinating groups of cells, like in the marine snail aplysia, where they release ink all at once. And it's not just electrical signals. Sometimes small molecules, metabolic signals, can pass through too.
Okay, super fast, direct connection. But you said chemical synapses are more common.
They are, yeah. Much more common, especially in complex brains like ours. They allow for more complex behaviors, more flexibility.
So what's different about them?
The main difference is there's no direct connection. There's a physical gap, the synaptic cleft tiny, maybe 20 to 40 nanometers wide, but still a gap.
So the electricity can't just jump across.
Nope. Instead, when the action potential arrives at the end of the sending neuron, it triggers calcium channels to open. Calcium rushes in. This calcium influx is the signal for tiny sacs called synaptic vesicles, which are filled with chemical messengers, neurotransmitters to fuse with the cell membrane. They then release these neurotransmitters into that synaptic cleft. The chemicals diffuse across the gap.
And dock onto the receiving neuron.
Exactly. They bind to specific protein receptors on the postsynaptic membrane, and that binding is what causes a response in the receiving cell, often opening ion channels there to start a new electrical signal, or sometimes triggering other changes.
Huh. So electrical signal turns into chemical, crosses the gap, and turns back into electrical. Or something else. That sounds slower.
It is. There's a definite delay, the synaptic delay, at least 0.3 milliseconds, often more like 1 to 5 milliseconds, sometimes longer, because of all those steps. Calcium, vesicles, diffusion, binding.
So why the trade-off? Why use this slower, more complicated system?
But it comes with huge advantages. One key thing is amplification. Think about it. One action potential might trigger the release of neurotransmitters from one vesicle. That single vesicle contains thousands of neurotransmitter molecules. And often, it only takes, say, 2 neurotransmitter molecules binding to open one ion channel on the receiving side.
I see. So one small signal coming in can cause thousands of channels to open on the other side.
Precisely. You get massive amplification. A small input can have a big output. That's crucial. Plus, chemical synapses offer much more variety in the response.
How so?
Well, there are different types of receptors. Ionotropic receptors are basically ion channels themselves. When the neurotransmitter binds, the channel pops open directly. Very fast response, milliseconds. Good for quick reflexes. But then you have metabotropic receptors. These aren't channels themselves. When a neurotransmitter binds, they kick off a cascade inside the cell, often involving second messengers like CAM-MP. This leads to slower but much longer lasting changes, seconds, minutes, even longer. They can alter the cell's metabolism, change gene expression. This is really important for things like learning, memory, modulating mood processes that need sustained effects.
Wow, so it's not just about speed. It's about the richness, the complexity of the conversation, fast signals, slow signals, amplification, modulation. The brain uses it all.
Exactly. It's an incredibly versatile toolkit.
Which brings us to another amazing aspect. This whole system isn't fixed. It's constantly changing, adapting, neuroplasticity. For ages, people thought the adult brain was pretty much set in stone.
Right, the old dogma was once you're grown, it only goes downhill, basically, except for maybe some degeneration.
But that's been completely overturned, hasn't it?
Absolutely. We now know the brain has this remarkable ability, neuroplasticity, throughout life. It can rewire itself, form new connections, strengthen or weaken existing ones. And in some areas, like the hippocampus, which is crucial for memory, it can even generate entirely new neurons, neurogenesis. It's constantly adapting to experience.
That sounds incredibly positive. Learning, memory, adaptation. But does it ever go wrong?
It can, unfortunately. Neuroplasticity also has a dark side, particularly when it comes to chronic pain. Sometimes these adaptive changes actually contribute to the problem.
How does that work?
Well, think about conditions like secondary hyperalgesia, where something painful feels even more painful than it should, or allodynia, where even a light touch becomes painful. These can involve neuroplastic changes. Often there's a process called central sensitization. If you have ongoing pain signals coming from an injury, say the neurons in your spinal cord and brain that receive those signals can become hyperexcitable.
The volume knob gets turned up.
Kind of, yeah. Their threshold for firing lowers. They start responding more strongly, and sometimes even respond to inputs that wouldn't normally cause pain. If the underlying cause, like inflammation, persists, these changes can become more permanent, structurally embedded. That's when you get fixed central sensitization, a hallmark of many chronic pain conditions like fibromyalgia, some types of chronic back pain, migraine, even phantom limb pain.
Wow. So the brain's attempt to adapt actually makes the pain worse, or creates pain where it shouldn't be.
In those cases, yes.
Yeah.
We even see physical changes, like shifts in gray matter volume in certain brain areas in people with chronic headaches, for instance, or nerve injury leading to a loss of inhibitory neurons, the ones that normally help quiet things down,
Hmm.
or fewer opioid receptors, making pain harder to manage.
And medications themselves can play into this plasticity, can't they? You mentioned opioids earlier.
Right. Repeated opioid use can actually induce neuroplastic changes in the spinal cord that contribute to tolerance needing more drug for the same effect, and can make certain types of pain, like neuropathic pain, even harder to treat down the line.
And with migraines.
There's evidence that overusing triptans, a common migraine medication, can lead to something called latent sensitization. It doesn't necessarily cause headaches directly, but it might make the person's brain more susceptible to their usual triggers. It primes the system, in a way.
It's a really complex interplay. But if plasticity can cause problems, can we also harness it for treatment?
That's the hope, and a huge area of research. For instance, there's a strong hypothesis that some antidepressants might work, at least partly, by promoting neurogenesis, stimulating the birth of new neurons in areas like the hippocampus. And researchers are actively looking for disease-modifying agents, drugs that could specifically target and maybe even reverse those maladaptive plastic changes involved in central sensitization, perhaps combining different drug types to tackle the problem from multiple angles.
So intervening in the plasticity itself.
Exactly. And it underscores why clinical guidelines often emphasize treating acute pain really aggressively. The idea is to prevent that initial barrage of signals from kicking off the cascade that leads to chronic, embedded central sensitization in the first place. Stop it before it starts, if possible.
It really shifts the perspective on treatment. Okay, from the brain's own adaptability, let's leap to something that sounds like science fiction. Actually controlling these neural signals with light. Optogenetics.
It does sound like sci-fi, doesn't it? But it's very real and incredibly powerful. It's basically a technique that blends genetics and optics using light to control the function of specific proteins within cells.
And didn't Francis Crick, predict this way back?
He did. In 1999, Crick actually wrote about the need for a method to turn specific neurons on or off in the brain using light, suggesting molecular biologists should engineer light-sensitive cells. It was remarkably prescient.
And they did it. How does it work, basically?
Well, the early applications focused on inserting light-sensitive ion channels into neurons so you could shine light and directly make them fire or stop firing. But the second-generation tools are even more sophisticated. Now, scientists can use light to control all sorts of protein functions, things like light-induced dimerization, making two specific proteins stick together only when the light is on, or oligomerization, getting several to link up, or even photo-uncaging, where light releases an active molecule that was previously trapped.
So you could precisely switch specific molecular interactions on or off.
Exactly. They use light-sensitive protein modules, like LV domains, CRY2 or PHYB. You genetically insert these into the cells you want to study, attached to the protein you want to control. Then, depending on the system, shining blue light or red light or far red light triggers a specific action protein's binding, changing shape, releasing something.
And the big advantage is the precision.
Unbelievable precision. You get spatiotemporal control. You can turn things on or off in specific cells at specific locations at precisely the moment you choose, just by aiming the light. And it's often reversible. Turn the light off and things go back to normal. It overcomes so many limitations of older methods.
I could see how that would be revolutionary.
Yeah.
What are some examples of what scientists are learning with this?
Well, it's huge in developmental biology, which actually gives us great insights into how complex systems like the brain get built. For instance, studying fruit fly embryos. Researchers used optogenetics to control a key signaling pathway called ERK. They found that what determines a cell's fate isn't just if it gets the ERK signal or for how long, but the total cumulative dose of the signal over time, a really subtle thing they couldn't easily see before.
That's fascinating. So history matters to the cell.
It does. Another example. They tagged a protein called bicoid, which helps pattern the embryo, with a light-sensitive tag, CRY2. This let them see how cells respond to changing levels of bicoid over time, showing that cells destined for fates needing high bicoid levels actually need longer exposure to that signal.
And you can even control cell behavior directly.
Oh, yeah. In zebrafish embryos, they've used a system based on PHYB to control proteins involved in cell polarity, basically, which way is up for a cell. They can literally use red light to turn polarity on in specific cells, making them behave differently, and then use far red light to switch it off again, back and forth.
Wow, like remote controlling development.
It's getting close. It's enabling what some call synthetic morphogenesis, not just observing development, but actively building or reconstructing parts of it. A great example is inducing tissue folding in fruit fly embryos. Normally, specific cells constrict to fold the tissue inwards during gastrulation. Using optogenetics, researchers could trigger that constriction machinery, Rho signaling myosin activity in cells that don't normally do it just by shining light on them. They can actually make the tissue fold in new places, controlling the shape and timing of the fold. It's moving beyond watching or breaking things to actively guiding biological construction.
That is incredible. And you can absolutely see the parallels. If you can guide tissue folding, maybe one day you could guide neural circuits to repair themselves or understand exactly how missteps in communication lead to disorders.
That's the long-term vision, absolutely. These tools give us unprecedented power to dissect and potentially manipulate the very processes we've been talking about.
What a journey today. Seriously, we started with that single neuron, that all or none electrical pulse,
The foundation laid by Hodgkin and Huxley.
then bridged the gap with synapses, both the super fast electrical ones and the complex, versatile chemical ones.
Allowing for everything from reflexes to learning.
Then saw how the whole system is constantly changing, adapting through neuroplasticity for better or sometimes for worse, like in chronic pain.
Yeah, that double-edged sword.
And finally, landed on optogenetics, these amazing light-based tools that let us actually get in there and control the conversation.
It really ties together, doesn't it? Understanding those fundamental mechanisms, like how ions cross a membrane, is exactly what allows us to develop sophisticated tools like optogenetics. And connecting it all, that deeper understanding is what lets us tackle really tough problems like chronic pain, not just by masking symptoms, but by potentially targeting the underlying maladaptive changes in the nervous system. It opens up completely new therapeutic avenues.
It really does. Which leaves us with a pretty profound thought, doesn't it? As we gain this incredible power, this precise control over the electrical and chemical signals that, define life itself, from how an embryo forms to how we think and feel. What does that mean? What new possibilities? But also what new ethical questions arise when we can understand and maybe even start to engineer the living brain? Something to think about. Thanks for diving deep with us today.